Hello boys, welcome back to another video tutorials, and this session is meant for class 12th biology boys, and this is for 5th October 2020. So we are into the revision of chapter number 5, principles of inheritance and variation. If you recollect from your memory, in our previous session we were talking about inheritance of one gene at a time. We call it as monohybrid cross, and we discussed what is the importance of that monohybrid cross. As I said, we all have the appreciation, the entire world appreciates Gregor John Mendel, the father of genetics, for whatever he has done hundreds of years back, even today holds good. That is the greatness of Mendel. So as I said yesterday, Mendel called genes as factors. Of course, at that time, not much of technology, not even hand lens. Now at least, you are all lucky that you are seeing under compound microscope. We are making an object 1,000 times bigger and seeing. But during Mendel's time, there was not even hand lens. So Mendel did all the experiments based on just one beautiful character, that is observation. So Mendel called the factors that passes through gametes from one generation to next generation. Now we are calling these factors as genes, right? And thanks to the genes for the hereditary characters that we possess. And nowadays it's called as genes, unit of inheritance. Genes that code for a pair of contrasting traits are known as alleles. Yesterday I said capital T, tallness, pure tallness, allele, small t, dwarfism, shortness, alleles, capital T, small t, heterozygous combination, again allele. So a pair of contrasting traits and the genes responsible for that is alleles. So alphabetical symbols, yesterday I said, do not think that there will be a gene called capital T, right, or small t. For our convenience sake, we are using the alphabetical symbols. So they are used to represent each gene, like capital letter capital T for gene expressed in F1 generation, and small letter small t for other gene. Mendel also proposed that true breeding, tall and dwarf variety, allelic pair of genes for height is homozygous, similar type of genes, capital T, homozygous, whereas the capital T, small t, that's heterozygous. So whether it is homozygous alleles or heterozygous alleles, they make genetic makeup of the individual. So we call it as genotype. So what is genotype? It is the gene combination and the gene ratio or genetic makeup, because of which every organism expresses its physical appearance. So whatever we appear today physically, it's because of our genotype, and whatever we appear physically, that's our phenotype. So phenotype is the physical appearance of an individual because of genotype, and genotype is the genetic makeup of an individual, which helps individual to express its phenotype. So the hybrids which contain alleles which express contrasting traits are called heterozygous. I said capital T, small t, homozygous. Here capital T, small t, different genes, so heterozygous. So the monohybrid ratio of F2 hybrid or F2 generation is 3:1 phenotypically. Yesterday we said 3 tall, 1 short. And genotypically 1:2:1, 1 pure tall, 2 hybrid tall, 1 dwarf. And I said this is universal. And to know about the genotype, we can do a small test called test cross. Test cross is the cross between an individual with a dominant trait, dominant trait, and recessive organism, in order to know whether the dominant trait is homozygous or heterozygous. Like for example, we said capital T, whether it is this way or heterozygous. capital T, small t. So whether the dominance is because of these two homozygous alleles or because of the presence of capital dominant gene. So here he is taking an example, homozygous recessive, the pod colors WW white, okay, and homozygous recessive again here. Now the combination what we are taking is, the cross is, what is the cross? An individual with a dominant trait and a recessive organism. So dominant one is pink, so the WW, and this dominant trait is combined with this white. So there is capital W, small w. Now when we cross this, all flowers are violet. Okay, all flowers are violet, as you are seeing in the Punnett square. That is because of presence of, you can see capital W, and the unknown flower is homozygous dominant. So all the flowers will become violet because of the presence of dominant gene. So homozygous dominant. Whereas on the other hand, the dominant phenotype, this of course why question mark, we don't know what is the genotype of it. So we are doing the test cross. So when we cross this, half of the flowers are violet, as you are seeing, because of the gene combination capital W, small w, and half of the flowers are white because recessive gene is present, small w. So if 50% of the F2 generation or 50% of the filials F generation is 50-50, that is 50% violet and 50% white flowers, then the unknown flower is heterozygous. So whether it is heterozygous or like this, heterozygous, and this is homozygous. So this is the purpose of test cross. So again, test cross is a cross between an individual with a dominant trait, okay, with the dominant trait. This is the dominant trait, okay, violet, and a recessive organism. This is recessive organism. In order to know whether dominant trait is homozygous or heterozygous. Now I said yesterday, based on the experiments that Krieger John Mendel did, he proposed 3 laws. Those laws are called laws of inheritance or principles of inheritance. So based on observations of monohybrid cross, Mendel proposed 2 laws. Yesterday I said law of dominance. So law of dominance states that characters are controlled by discrete units called factors. Of course now we call gene. Factors always occurs in pairs like capital T, capital T. In dissimilar pair of factors, one member of the pair dominates the other. Like we said, capital T, small t. Capital T is dominating over the small t. This is what he said. And the dominance and recessive. When factors, that is alleles, express itself in the presence or absence of its dominant factor, then it is called dominance. So the capital T and capital T, no doubt pure tall, it will express both genes are dominant. Even if one gene is present, it expresses tallness, then we say this is dominance, capital T is dominance. Whereas recessive, it can only express itself with the absence of or its recessive factor allele. Here, the small t will express itself only when it is homozygous condition. Then only this will express dwarfism. Whereas any other combination, this recessiveness cannot be expressed. And dominance, it forms complete functional enzyme that perfectly expresses it. It's because of the enzyme. It forms incomplete defective enzyme which fails to express itself when present with its dominant allele. So in heterozygous condition, it cannot. And second law that he proposed based on this is law of segregation. Alleles do not blend and both the characters are recovered during gamete formation as in F2 generation. What is segregation? Separation. Separation of the genes in the gametes. So during gamete formation, look at this, during gamete formation, traits segregate traits get separated from each other and passes to different gametes. Homozygous produce similar kind of gametes, but heterozygous produce different kinds of gametes with different traits. And here we have talked about that law of separation, that is capital T, one comes to one zygote, another comes to another zygote, they get separated. Similarly, capital T, small t, we said this gets separated, so you got 4 gametes. So this is how they separate each other during gamete formation. And in the law of dominance, there are some exceptions like incomplete dominance. Incomplete dominance is wherein the dominant gene will not express its dominance completely. So there is something called hybridization. So it is post-Mendelian discovery. This is not during Mendel's time. The neo-Mendelists or neogenesists have found out. Incomplete dominance is the phenomena of neither of the 2 alleles being dominant, so that expression in the hybrid is a mixer. This is important, fine mixer, or intermediate between the expression of the 2 alleles. So you must remember Snapdragon, Mirabilis jalapa. Again, this is one more question. Snapdragon is Mirabilis jalapa. There are 2 types of pure breeding plants, red flowered and white flowered. Okay, red and white. On crossing the 2, F1 plants possess pink flowers. Look at this, we are getting a new character. Normally, law of dominance, red color capital R versus when we say white flowers, we have to take corresponding alphabet. Don't take W, okay? Small w, don't write. Instead, you have to write small r, right? And in law of dominance, what happens? In F1, this is parental generation. In F1, what happens? Capital R, small r versus capital R, small r. Now, this is F1 generation, right? So, this is the combination and this is the combination, right? So, what is the phenotype of this? If it is a law of dominance, because of the presence of capital R, this should be red flower. But what's happening in Snapdragon, Mirabilis? It's becoming pink instead of red. This is the new character, new variety. Why this pink color is coming? The pink color is appearing because this dominant gene is no more dominant. It is only incomplete dominant, means something like 75%. And this small r is able to express the remaining 25%. So that makes, thanks to the variety that is formed. So on crossing the 2 F1 plants possess pink flowers, as I said. On selfing them, F2 generation has 1 red, 2 pink, 1 white, 1:2:1. The pink flower is due to incomplete dominance. Now, the same thing in the form of Punnett square. So, you got first capital R, that is a red flowered snapdragon crossed with white flowered snapdragon. So, smaller genotype. So, the combination, law of segregation. Capital R from this red flower, small r from white flower, you are having capital R, small r. This is pink flower. And in F2 generation, we are going to write this capital R, small r. And on top, capital R, small r. Now the combination is capital R. So what do you expect? Homozygous dominant, red flower. Okay. And this zygote combines with this small r. So capital R, small r, here law of dominance is not working, law of incomplete, so pink flower. And so as this capital R, small r, so pink flower, incomplete dominance, and of course small r, white. So you are getting 1:2:1 incomplete dominance. No more 3:1. See, phenotypic ratio is 3:1. But I said it is universal, but some exceptions are there, like Mirabilis jalapa, 1:2:1. So 1 is genotype, if you write capital R, 2 is capital R, small r, and 1 is small r. So capital R is red flower, and capital R, small r is pink, thanks to incomplete dominance, and homozygous recessive is white. So this you have to remember. Exception to law of dominance is incomplete dominance. So further we proceed with some more interesting experiments like this. What is that you have to do? Keep one 200-page notebook, write all the revision notes, plus I will be posting the NCERT question answers, and that also you write. So for every chapter, starting from chapter 1, you have to write revision notes. At the end of the revision notes, you write question answers. After that, like last week I posted MCQs, tomorrow I am posting another 50 questions, answer that. This is followed by every chapter I will be posting MCQs as well as assertion and reasoning questions. So I am sure you got now videos of the chapter, right? Second, revision notes of the chapter. Third, NCERT question answers. Fourth, MCQs. Fifth, assertion and reasoning, and whatever neat UPSC NDA questions of the chapter. I think with that you must be perfect, right? All the best.